We'll get into the next presentation. The presentation is on evaluation of yield variation in Sri Lankan traditional rice, especially Kaluheenati. There are two presenters, one WHDU Pushpakumari and S. Geekiyanage.
Good afternoon, all of you. Actually, evaluation of a variety for potential of a yield. It's provide much information for the breeders to develop new varieties for the country requirement. It is an essential need. So my research based on evaluation of yield variation in Sri Lankan traditional rice, Kaluheenati. So why rice is such an important crop to the research population, research world? Why? Yes, rice is the world's second largest produced cereal after maize, and it is the staple food in most of the Asian countries, including Sri Lanka. And do you have any clear idea about rice germplasm in Sri Lanka? Do you have any? Yes. Actually, in Sri Lanka, we have rich rice germplasm. There are nearly 2,000 traditional rice accessions which are available in National Gene Bank in Plant Genetic Resource Center, Gannoru. But the thing is, most of the traditional rice varieties had been replaced by regular farm fielding of new improved varieties introduction with the Green Revolution. The main thing is, these traditional rice varieties, they are known to be a very good medicinal properties, nutrition properties, and also very good pest resistant ability. Not only that, and there is a trend now. These traditional rice varieties can be used and integrating into the farming system in agriculture for the desirable character that incorporated with the organic farming. But the main problems, main barriers in counting of these traditional rice varieties reintroduction to the farmer field. What? Yes, it's relatively low yields, and also most of the traditional rice varieties long age varieties. This is the problem. So Sri Lankan traditional rice variety Kaluheenati is known to be a very good medicinal and nutritional properties, as well as it is cited as a short age variety. But nobody have ever been studied about this type of such an important variety for the yield performance in relation to yield variations that nobody ever been studied yet. So, according to this situation, in Sri Lankan context, to solve these problems, we have formulated the objectives. The first objective, the identifications of yield variation of Kaluheenati accessions. Second one, that determinations of the relationship between days to flowering and yield of Kaluheenati accession. To accomplish with these objectives, we used 26 accessions of Kaluheenati from Plant Genetic Resource Center, Gannoru, and they were grown at Rice Research and Development Institute, Batalagoda. The experiment set out with 4 replicates in randomized complete block design during Maha season in 2013. So we have noted in the first problem of the justification, we don't know the variations of days to flowering for the introduction to the farmers. So we have collected the data on days to flowering, then yield components. Under yield components, we have noted total panicle weight and 100 grain weight, grains per panicle, and field grain percentage. All the data were analyzed by using SAS, and also the mean separation was performed by Lankan multiple range test, and correlations of coefficients calculated by Minitab 15 version. Okay, let's look on the results and discussion part. Yes, the first objective, the first problem is this phenomena, the flowering time variation. Study about the flowering time variation. So you can hear the most of the Kaluheenati accession were flowered during 75-79 flowering age group, and also variation from 61 to 85. This is about flowering time. Let's move about the yield component. First yield component is total panicle weight. Here accession number 2087, 4091, 5191 had significantly different from others because the highest total panicle weight recorded by these accessions. And also accession number 2100 was recorded significantly lower total panicle weight. This is about total panicle weight. Actually, yield of a rice is a multi-trait character. So evaluate the yield, we can use these yield components. Then 100 grain weight. The accession number 4090 gave the significantly highest mean of 100 grain weight, and also accession number 6713, 4086, 6232 and 4740 recorded the lowest value of 100 grain weight. And the main thing, at a special results of this analysis, there was a significant positive correlation between days to flowering and 100 grain weight. This significant at 5% probability level. Already I have discussed about days to flowering, yield component, under yield component, total panicle weight and 100 grain weight. Let's move about how about the grains per panicle. Accession number 5191 had produced significantly highest mean of grains per panicle, and the accession number 4253 significantly different from other accessions, other Kaluheenati accessions that recorded the lowest means of grains per panicle. Let's move to the field grain percentage. The accession number 6713 and 4740 had produced significantly highest mean of field grain percentage, and also accession number 2090 was recorded the significantly lowest field grain percentage. Actually, this is about the yield component. The variations and what type of accessions significantly different from the whole accession conserved in Plant Genetic Resource Center in related to Kaluheenati variety. All the research can be summed up and can be concluded. The highest mean of total panicle weight, 100 grain weight, grains per panicle and field grain percentage were recorded by accession number 2087, 4090, 5191, 6713. Actually, according to the results, I think you can be observed how varied this yield among the accessions, among one variety. The total panicle weight, you can see the value, the variation from 24 to 6, and 100 grain weight, grains per panicle, field grain percentage, and days to flowering. The variations, you can see the big diversity among the accessions of one variety, and also there is a significant positive correlation between days to flowering and 100 grain weight. So this is information, this information and knowledge results would be a good raw material for the future breeding program in Sri Lanka for variety development for the increased yield. Actually, finally I would like to acknowledge National Research Council for the financial assistance, and the Plant Genetic Resource Center, the provisions of the Kaluheenati accessions. These are the references, and thank you all of you for your kind attention. Thank you very much.
I'm sure you will have a lot of questions to ask. Probably you all are waiting till I ask the questions, because I am from the Ministry of Agriculture. Yes, in very interesting work. Very interesting work, because we really have a whole lot of accessions in our Plant Genetic Resource Center. When we take one traditional variety, we have more than 30 or 40. Sometimes when it comes to Suandal, we have something like 38 accessions. So we don't know which one is the real Kaluheenati. And when it comes to Kaluheenati, we know the Behetheenati is also there. It has very low yield. The main issue in these traditional varieties is low yield. More than that, they are mostly susceptible to blast. So it can give a good yield in one season, but when there is blast, you may not get any. And also most of these varieties are photosensitive. So those are the issues why we don't use them anymore. But there is a potential because of, I'll use the word therapeutic value rather than using the medicinal value. Medicine in food is questionable, medicinal value. So therapeutic value, it's not yet proven in most of the traditional varieties. But what we have done is very important. I'm very happy that we are doing that. If we don't have this kind of information, we can't use it in our future breeding programs. Of course, there are the problems of using them in the usual breeding program. There are the barriers, but then if we don't have this kind of information, we don't know which variety to use. Just by looking at the quality characteristics, we can determine the varieties. So it's very interesting work. Thank you very much. We show our appreciation once again.
Thank you very much.